MasterMixes are your friends in the lab, and spreadsheets to make MasterMixes are your friends before you do the lab. And so this is just a spreadsheet showing a couple of examples of strategies for preparing MasterMixes or for calculating how to prepare MasterMixes. So first off, what's a MasterMix? It's basically where you have samples that have shared components and unique components. And you premix the shared components so that you minimize the amount of pipetting you have to do. So you can premix the components that are shared between all the samples and that are stable, and then just add the unique parts to them. And so here I have two kind of main strategies for doing this. One is that I figure out how much volume I need, and then prepare to make a volume that's bigger than that. And I calculate the fraction of like each component's fractional volume kind of. So it makes up this much of each reaction. So it'll make up this much of each master mix. And then I calculate based on that volume. Another strategy is to say, okay, I need this many reactions. I want to make this many reactions worse so that I have enough for extra. And then I prepare by multiplying the volume per reaction by the number of reactions I want to prepare for. Both have their pluses and their minuses, and so this is either way you're calculating so that you're making more than you need, but here it's based on how much volume you want to prepare, and here it's based on how many reactions worth you want to prepare. So I use different ones at different times, and I have shown you examples of how you could use both of these in a spreadsheet. So you'll have to change things depending on your needs, but here's just the basic walkthrough. So over here, I have these columns where I have my stock concentrations and my desired concentrations. You can have something similar to this where you then direct the cells to go to the well that you want. And note that I have dollar signs here because the dollar signs are going to tell it this is an absolute reference. And so it's going to go back to this single reaction volume each time, even if I were to copy and paste this into a different cell. Much more on dollar signs and that sort of thing in the graphics. So this is part of a bigger spreadsheet lab calculations example spreadsheet that I shared, and I have a longer video and walkthrough on. But so this is just one page of it. There's a bunch of other stuff, and I also will direct you to some more MasterMix calculation examples. But going back to this one, so we have a per reaction volume and we say, okay, well, we want this much of each of these components per reaction. And so these can be a set volume that you then adjust. So things are in blue are things that you're going to adjust based on your experiment. Well, I mean, you also have to adjust the components that you want in here, but that's why it's really important to actually know the math that's going on behind all this. So what we're doing is basically some simple C1 V1 equals C2 V2 type stuff. And so review those concepts if you are confused about that. But basically what it's doing is it's going to go and it's going to calculate our V1s for each of these reaction components based on their initial and final volumes. Or again, some of these I just put in based on what I know I'm going to put in the reaction. Okay, and then you need to figure out, okay, well, how do I get it to that desired volume? Often this is going to be with a buffer or water. And so here I have a buffer and it's going to, the amount of buffer per reaction is going to subtract the volumes of each of the components. So that is per reaction. And now I want to say, okay, well, I have 6 reactions I want to do. Which of these components can I premix? I don't want to premix the enzyme because one, that's going to be different. And two, I want to make sure it stays stable. Oxaloacetate, that's a pain in the butt. And actually, that's how I'm going to start the reaction, but it's really unstable. And so I want to add it last. But I can premix the NADH and the buffer. And so let's do that. I know that per reaction, I have 10 microliters of NADH and I have 970 microliters of buffer. So if I mix those together, I have 980 microliters of MasterMix per reaction. And so you can see that this is basically just adding those up. I want 6 reactions. So I know I want to do 6 reactions, but I also know that I want to make extra. One is that there's some volume loss during transfers. Another is that, so if I prepared for making 6, I probably wouldn't be able to make 6 because I would have lost a little more. And so I probably would have only been able to do 5 reactions with a little less left over on this for the 6th one. Another is I'll probably have to redo a couple of them for various reasons, pipetting errors, not starting the machine quick enough, having to figure out what's the right enzyme dilution. So I want this to be higher than 6. And so I want to make more than 6 reactions worth. And so in the second strategy, I say, okay, well, maybe I'll just make enough for 10 reactions. Another way to do this, though, is to just say, okay, what's a convenient volume for me to make? Convenient volume would be 10 mils, right? So this would be 980 microliters per reaction. If I multiply that by 6, then I end up seeing that I need to get 5.88 milliliters. Here it's divided by 1,000 because this was in microliters and this was in milliliters. If this were in milliliters, then I wouldn't have to divide it by 1,000. But again, this is just where you kind of have to change things depending on your experiment. And so it's important to know what's going on. So I say I want to prepare 10 milliliters. And now I say, okay, well, how much am I going to need of the NADH and of the buffer per in that? And so what I'm going to say is, okay, well, what fraction of the master mix is the NADH and what fraction is the buffer? So remember that the NADH was going to be 10 microliters worth of the total reaction. So it would be a tenth of the total reaction volume. But the master mix is only going to be 980 microliters that we put in each sample. And so it's going to be less. It's missing those other components. So it's going to be more than a tenth. And so if we actually go and calculate this, we can see that if we multiply we divide 10 microliters per in our master mix divided by 980 microliters of master mix, and then we multiply that by the 10 milliliters, we'll see that we need 0.102 milliliters, which is 102 microliters. So you also want to get really good at kind of just moving those decimal places in your head. So to go from milliliters to microliters, multiply by 1,000, we take our decimal place, 1, 2, 3, 102 microliters. Now, what about our buffer? Well, our buffer, we do the same thing, except here we're going to direct it to say, okay, we need 970 microliters of buffer per reaction, and we have 980 microliters of the master mix per reaction. 970 times divided by 980 times 10 milliliters, and we find we need 9.898 milliliters. And so that is how you do it based on the volume. And so I said, okay, well, 10 mils would be a convenient volume to prepare. Let's prepare 10 milliliters. Okay, strategy number 2. In strategy number 2, I prepare based on the number of reactions worth I want to make. And so similar things are going on. We said, okay, well, we want the same reaction that we're going to do. And so I'm able to just kind of like copy and paste that. If I had put dollar signs, that would then direct it so that it's going back to the stock concentrations and to the desired concentrations that I have in that initial column. Be careful if you don't put the dollar signs, then when you go and you copy things over, it'll be directing it to the wrong cell. So you want to make sure that you are checking to make sure that you're always directing to the cell you want. So it's always a good idea to click in the cell, click on the equation bar, and then it should highlight the cells that it's directing you to. So this part was the same. The numbers are just a little different because now, or I guess the letters are different because now we're in a different column, but I can do the same sort of thing. So here I still want to make 6 reactions worth, but now instead of preparing for 10 milliliters worth, I want to prepare 10 master mixes worth, or 10 reactions worth. So you can see that the volume's a little smaller because, well, here, if I have the 980 microliters per reaction, and I multiply that by 10 reactions, that's not going to be the full 10 milliliters. Instead, it's only going to be 9.8 mil. Okay, so now let's go and figure out how much of each of these components I need. Now it's a little easier. I just need to take the per reaction volume, multiply it by the number of reactions it's worth to prepare, and voila, I figure out these values. So apologies before, I had forgotten to change this to say microliters instead of milliliters. And so remember over here, I had it dividing by 1,000 so that I would end up in milliliters. Here I don't, and so my answer is going to be in microliters. And again, if I want to go the other way now, to go from microliters to milliliters, I divide by 1,000, 1, 2, 3, and then so like 1, 2, 3, I would see that this would be 9.7 milliliters. Spreadsheets can be really helpful. What's even more helpful is learning how to code. And so if it was just me, I might be doing these calculations with the custom Python script or something. But when you're working with undergraduates, as well as just when you want something that you can easily see, and if maybe you don't know coding, all this stuff, spreadsheets are a really great tool. But I do recommend learning Python and trying to write functions to do these things for you. But anyway, going back to the spreadsheets, one thing to be a little aware of is be careful when you're copying and pasting. So always be checking, make sure that the values before you copy them are the same as the values after you copy them. And so when you just want to paste the values, what you want to do is do Command-Shift-V. And typically what I'll do is I'll have like a to prepare tab. And what I'll have is I'll have the final volumes without all of the calculations. And this is going to let me see really quickly what I need to do. And so what I could do is if you just copy and then Command Shift V, you'll paste just the values. And now maybe what I'll say is, okay, well, I'm going to have that. I want to know how much I need to make per reaction. I want to know how much I'm going to be making, and then I want to know how much of each thing I'm going to need. I might go in here and say, okay, well, this is 9,700 microliters. Let's change that to milliliters. And so if I just divide that by 1,000, and so I could just move my decimal place 1, 2, 3, and I say, okay, well, I'm just going to rewrite this as 9.7 milliliters. And that is then what I go and I have this in my tab and I know exactly what I need to mix together, how much of it to add per reaction, and how much total volume I'm going to need. And this is going to help me say, okay, well, maybe a 50 mil conical tube would be a great thing to make this in. Having a simplified kind of what to make when is really helpful in the lab, and then you don't have to worry about being boggled down by all these calculations because you did the calculations ahead of time and now you know exactly what you need to do in the lab. So, hope this was helpful, and I will post links to this as well as to the other MasterMix calculation examples I have. And so, those ones are all done in the form like the second one, but I also do sometimes use this first strategy where I prepare a certain volume's worth rather than preparing a number of reactions worth. I still think it's useful to see what's the final volume you're going to have when you're doing it, like by reaction numbers, because that helps you know what type of like plasticware you might need, like what type of tubes and things you were going to prepare this in, as well as allows you to see like, uh-oh, it's my volume isn't what I expected. Maybe I did something wrong. And make sure that whatever you're doing, you are checking to make sure that your answers are reasonable. And so that's just making master mixes really, really helpful in the lab. And so hope this helps you strategize when you're setting up your reactions and that sorts of thing.